The Self of Endogenous Viruses, a presentation by Professor Gershom Zajcek, Faculty of Medicine, Hebrew University of Jerusalem. This presentation is a continuation of a series discussing the Gaia Genesis in Gaia. Gaia is a superorganism in which life evolves, and I am describing its evolution, which is known Gaia Genesis. In a previous presentation, I showed that innate immunity was created in single cells in young Gaia. So here is the explanation how these single cells created innate immunity. So here you have a prokaryotic cell, its DNA, and a Provirus, which is a set of genes in its DNA, and this set of genes control the production of virion, and they store also the self of the cell. So here is the example. We are just observing a cell which produced virions, and the virions are now exiting into the environment and attempt to infect other cells. So the outcome of this attempt there are two possibilities. If the cell has the same self like the virus will attach to a receptor and following uncoating interact with the provirus. But if the cell has a different self, nothing will happen. Virus will not attach to the receptor, and there will be no uncoating because the provirus blocks it. So provirus protects cell against non-self, and this is innate immunity. So there's a process which I described, which describes the interaction between virus and cells, and so this is, I call it virus-provirus process. Virus attached to the cell, after uncoating, the virion or the genome is entering the cytoplasm, interacting with the provirus, and the provirus controls the formation of new virions, and the provirus stores also the self of the cell, and which means that the virions which are controlled by the provirus carry the self of the cell. So, some definition: self of cell is a set of genes stored in the provirus, and virus carries the self in its genome. And generally we are talking about cell families, and cell families have cells which the cells have the same self. Now it so happens that in the scientific literature, provirus is called endogenous virus or endogenous virus element. So an endogenous viral element is a DNA sequence derived from a virus and presents within the germline of non-viral organism. Germline DNA refers to to tissues derived from reproductive cells that become incorporated into the DNA of every cell in the body of the offspring. So we are talking about endogenous viruses, and here you have an example of a review of these endogenous viruses, which is called Connecting Recent and Ancient Viral Evolution. They write, the rapid rates of viral evolution allow us to reconstruct the recent history of viruses in great detail. This problem has been partially solved in the discovery of viruses embedded in host genomes known as endogenous viral elements. So, what are we talking about? We start as usual with Baltimore classification, which described 7 classes of viruses which may infect the cell, and they trigger the formation of provirus. Now, what is now new in this classification, that there are 7 classes of proviruses, and you can represent this relationship by the Baltimore process. And this Baltimore process, you have here the 7 classes, all interact with the cell to create 7 proviruses, and then these 7 kinds of proviruses control the messenger RNA in order to form the viruses. Now, this what I'm telling you now about this EVE is a modification of this Baltimore process. We are talking about endogenous viruses. These are 7 classes of endogenous viruses which interact with the cell and are manifested as endogenous viral elements. So, what does it mean in terms of our study of Gaia? That the provirus in this virus-provirus process is actually an endogenous viral element. So, conclusion, provirus blocks entry of non-self. Provirus blocking is innate immunity, and endogenous viral element is a manifestation of innate immunity. I hope you like this idea and enjoy.